I
Oh,
got you, Laura.
oh, nice job, man. Okay, beautiful.
Do we want to capture all that small talk at the beginning as well? Or
What,
we're
like
good without
redo
it?
the small talk?
Maybe we'll skip it. Proceed.
Okay. Um, yeah, so here's the agenda for tonight. In a second, we'll talk about some of the results that have come back so far. Um, And then Alex is going to speak about the sequencing process. So we'll talk about that second. And then we'll talk, this is kind of like in chronological order, kind of, except for the results. So we'll talk about the sequencing process, then from sequence to identification, and then understanding identifications and the comments you see on iNaturalist, what do they mean? And then lastly, sort of next steps and getting involved. I think you all are already involved in the project, but this is specifically kind of like getting involved in this back end stuff that we're going to be talking about tonight. So that is the plan. And I have one more slide here that's kind of an intro slide. I made this right before because I just had a few notes I wanted to share. So first of all, thank you to everyone for your collecting and participation and like enthusiasm. There's a lot of it, and it's really awesome, I think, seeing the project here in BC continue to grow and just like the potential of the magnitude of the size of this project, how, far we can take it and how much we're going to learn about fungi in the province is all very exciting. And thank you so much for all the organized packages that I've been getting recently. So I know in the last meeting I kind of had some requests about how they get sent in, and yeah, they've all just been a dream coming in recently. So thank you. And then I wanted to shout out Ben, who I believe is here, who spent some time making this website that does some data quality control checking for everybody. That is part of why the packages are coming in a dream, because not only are they physically organized, but all the data has been sort of um, checked for errors and duplicates and all that stuff. So thank you, Ben. And yeah, there's just, I think like it's hard to communicate on my end where I go into work and I talk to Brian, who some of you met last time, the professor that um, I'm working with, and all my lab mates and all these people, just the the interest and the potential in this project and how stoked people are, like even outside of mycology, for the sort of like template we're creating here that can be applied elsewhere and to other taxa. So yeah, I just wanted to say that because I think about it every day. So yeah, thank you all. And really the core of this, I guess, is the collectors. When I was talking to Steven the other day, he was like, yeah, in the future, like almost all of this stuff in the back end could be heavily automated, but the one thing that can never be automated is collecting mushrooms. So that is like, yeah, really the base and the most important part of everything. Sweet. Okay, so we can jump right into looking at some interesting results from the project so far. So we have had 200, I just checked, 213 sequences. Well, that's based off a certain timeline that I've applied. More than that that have come through so far. But anyway, over 200 sequences have been validated so far and had the identifications posted on iNaturalist. So pretty small sample size, but still lots of really, really cool records and interesting finds. So we'll talk about some of those. And I've put mine first here, and then I'll pass it off to some other folks. Okay, so I wanted to talk about this one. Cool for a couple of reasons. One, because it was found by a grade 7 student. So you can see the INAT account here. This is the name of the school. There's a teacher there who's a very keen naturalist, and she took her kids out collecting. They made about 30 collections, and we just have 10 of their sequences back. But actually, 3 out of their 10 sequences were BC firsts. So you can imagine these grade 7 students were very stoked. I actually went to their class to follow up and present and talk to them about it. And yeah, it was just awesome. They were like, they were very excited. They were like, wow, we were really locked in. They kept saying that, how locked in they were. So yeah, it was amazing to see them be very excited. This was one of their BC first. So um, there's two things going on here. We have just a Swillis. It's Swillis lakei, very common. And when we got the sequence, that's what they were collecting really, right? Was this Swillis. And when we got the sequences back, we had two sequences, one for the Swillis, which is kind of the host uh, mushroom here, and then one for this parasitic entoloma. Um, that is this white patch of tissue that you can see on the mushroom. So this observation that I've put into the slides is the secondary observation we created to make a new one for the sequence for this entoloma. So very interesting because it has not been recorded on INAT in BC before. It was really cool that we got two clean sequences back from this, one for the bolete and one for the parasite. And if you look up this species on INAT, you can see other observations where like the parasite, the parasitism is a bit more clear than even in this one. But yeah, just cool. And we've seen this on a couple observations so far. Like we're able to sometimes get a sequence back for like a parasite and a host, both fungal, which is awesome and interesting. So that's one I wanted to share. And then the other one is just the Stictus. So I really like Stictus. This one's on Arbutus. I collected a lot of them, and I wasn't sure if they were going to be like iNaturalist always suggests Stictus radiata or radiata. And I was like, oh, is that really what it's going to be? And I was also like, how well are these going to be sampled? They're kind of like embedded in the bark. And when I first was getting started collecting and people had all these questions for me, like, can I send in this? Can I send in that? Is this too hard to sample? I didn't really know how to answer. And now I basically answer like, if it's just a few things, like, let's try. So I'm trying all sorts of things in the lab this week that I don't know how well they're going to come back, but this one did, and it has a temp code Stictus CA01. So it's found first in California. It was the first one. And it wasn't even the best match to that, but it was on the same substrate on Arbutus. So I applied that for now. And I'm going to see because I have a lot more coming back. I'll be able to look at them a bit more closely. So those are two I wanted to talk about. And I will pass it to you, Luke.
Okay, yeah, so these are two recent validations that I made of Alora's collections. And I chose these two specifically as interesting because they both came up as needing temp codes. And now Alora just mentioned temp codes on the previous observation, so you might be wondering what good is a temp code or what is a temp code. So just to give you a background on that, so when validating a sequence of a mushroom, throughout the process, we're essentially presented with possible matches. Alongside those possible matches, there's a percentage figure of how close that DNA is. So in this case, both of these observations were a significant percentage away from that nearest available genetic match. Now, since because of that, we don't have any support to confidently put a specific species name to these, they each received a temp code. So you can think of it kind of as like a placeholder name for the time being. So you'll see temp codes with different initials, and those usually reference area. And that's kind of the format that we've adapted here as well. And we're simply adding DC and then a sequential number to name these essentially new finds. And now temp codes are just that, they're temporary. So the idea is that it's a placeholder for the time being, and eventually there's going to be further action taken. And that action could be anything from the mushroom being described as a new species. Different temp codes can maybe be merged together as we realize that they're actually closely related with each other. Perhaps a single temp code can be further split up as we learn that maybe there's actually multiple distinct species present within that temp code. Or maybe altogether a temp code could also be completely scrapped if, let's say, a mushroom that was described 50 years ago receives its first DNA sequence, and now all of a sudden all these temp codes match that already described species. So all that is to say that anything is really a possibility, and further genetic data will eventually clarify this bigger picture. But these are both really exciting finds, and Alora is the proud owner of BCO1 and BCO2, and I think also 3 and 4 now for Inocebi, if I'm not mistaken. And one other quick point I wanted to make here with these two observations is that they were both found in the southern Okanagan. And I don't know if folks are familiar with this area, but it's a really arid and dry environment, and it's not a place where most mushroom pickers go. And this is one of those things that I love about a project like this, and particularly MycoMap, is that things are being collected all over the province, not just in, prime second growth Douglas fir where we're all finding chanterelles and just find some other cool stuff on the side, but there's some really cool habitats and ecosystems being targeted, and we're pulling some really cool finds out of there. And a lot of the times, uh, specimens in these sort of not traditionally visited areas for mushroom picking are generally understudied. So we're going to start seeing some really cool stuff come out of there. Next slide, please.
Cool. And sorry, I forgot to like do any like introductions for other people that are talking, so I don't know if you will like want to say anything, but
Sure.
everyone that's sharing tonight too, part of the reason that you can see in these things like Luke did these validations himself, so he's been going over all the data we've been getting and you'll see more of that process later, but um yeah. So sorry, Luke, I'll switch the slide for you now.
All good. Maybe between the slides here, I can introduce myself. I'm
Yeah, sorry.
Luke. I'm based out in the U.S. in the Lower Mainland, part of the Vancouver Mycological Society and the Pacific Northwest Tea Council. And yeah, just super excited to see uh, all the stuff that's been going on with MycoMap in so little time, but really exciting to get these results back. Start to see all the IDs pouring in. Yeah, I'm going to chat about this one here as well. So this is a collection by Mitch Milgram, who I think I saw on the call here today as well. And I chose this as a notable one to chat about because of its resemblance to a really common, but also extremely variable mushroom. So this was originally identified in the field as Foliota terrestris, and that's a common Foliota that probably a lot of you have come across while you're out looking for mushrooms. And rightfully so. When I saw the observation before diving into the DNA and the results, I agreed totally. Um, but when we ran the DNA and looked at what the possible matches were, we got a really strong match to, I believe it was the type sequence for Foliota kodiakensis. Um, and that's a Foliota that's never been found in BC before. All of the observations on iNaturalist, I believe, were in Alaska, and a couple in Alberta, but never in BC. So as a BC mushroomer, the species was never even on my radar. This is the first time I heard of it. Um, I got this book recently in the mail, Noah's Mushrooms of Alaska, and sure enough, it's in there, and also talks about its striking resemblance to Foliota terrestris. So this is just a cool example of a discovery for our mycological community in BC that wouldn't really have happened without the help of that DNA. And because of this, we can really paint a clearer picture of our fungal diversity here. So I just thought this one was really awesome. Cool find there, Mitch. And I think, yeah, the next one is somebody else.
Let's
I
see,
think this is to
this
you,
one's mine.
Alex. Yeah.
Yeah. So this little hygroscopy, also hi, I'm Alex, I guess. I can introduce myself first before I talk about this. I live in the Kootenays in British Columbia, and I have worked for Mycota Lab in the last year or so, um, which is the lab that does a ton of the sequencing, specifically like all of the sequencing for MycoMap BC, and just other community sequencing projects and such. But now I'm back in Canada, which is nice. And I'll be talking more about the sequencing later. But right now, this little hygroscopy that I've got here, it doesn't look much different from other hygroscopies. It's orange, it's small, it's kind of gooey when it's wet. But what's interesting, and you'll see from that map there, is that it has only been found right here in the Kootenays and in where is that? Ohio and in Florida. And the temp code it was given was Florida 07. And I just picked this one because it is extremely interesting that a mushroom that was found originally in Florida made it all the way over here, has been found all the way over here. So who knows what's in between it? Who knows how it got here?
It's a
Uh,
snowbird. Snowbird.
right? Florida and Ohio and the like central Kootenays is such a crazy distribution. And that's why it's important to pick samples of things, even if it's something that you think you've seen a million times, or that looks no different from something else you may have found in sequence before, because maybe it's something from Florida that just looks the same as everything else. And that's the kind of data that we want to be able to have. That's all I really have to say about this hygroscopy. Is my next slide? Yeah, this is just a bigger version of the map. So yeah, you can see up near Vancouver and then Ohio and then down in Florida.
Yeah, I wonder
And
if it
then
occurs all the way throughout too, and we just don't know yet.
exactly. I wonder what the climate is in common between those three spots other than just being kind of wet-ish, maybe? Hygroscopy is just like wet anyway. And then the second one I have to talk about is Nectariopsis violacea. And you can see why it's called violacea because it's kind of purplish. And I just thought not only is this a fairly rare thing to find overall, I didn't show its whole occurrence map, but it doesn't come up that often. But this one is just a cool species because the thing that its substrate there is actually a slime mold. It's growing on Fuligo septica, and it parasitizes that slime mold. And I just thought that was neat.
Yeah, I
That's
love
about
it.
it.
I like
It's
the
not super interesting
background.
genetically. It just matches the type sequence and other observations for Nectariopsis violacea. But I just thought it was a neat, a neat example of something weird you can find in sequence.
Before I knew what this was, I was looking at it and I was sure something fungal was happening, and everyone else I was with was convinced I was handling dog poo. I was like, it was like an old one, but
Sometimes it's hard to tell.
it was indeed this.
Very fun.
Okay, over to you, Leanne.
All right, it's my turn. My name's Leanne Rattan. Same last name as Alex. Yes, I am Alex's mom. And I'm also in Castlegar, BC, where I am associated with the Kootenay Mycological Society, and I also work at Selkirk College in the School of Environment and Geomatics. So, oh, hi, Nathaniel. All right. So the one I'm going to talk about first is Pluteus hongoi. Um, so I first found this species before this observation in October of 2024. I actually found it twice on the same day, but the specimens each looked so different from one another and they were a fair distance apart. I didn't know they were the same. So I got two sequences back in October of 24. But then in 25 here in August, I found this Pluteus here. Um, and it was first ID'd by Stephen Russell, who runs the Mycota lab that does all of our sequencing. But at the time, he didn't upload a sequence, so I wasn't really sure whether I believed it. Can you give me the next slide, please? Given the distribution map, if you can see all the East Coast observations of this species, and then way over on the left coast up top in BC, there's my three observations in one in BC there. Um, then who was it? Ken Sanderson came along and gave the same ID, and he also posted the DNA sequence. And then I agreed with that. I said, okay, I guess Stephen knows what he's talking about. Um, so that was my first one. So now there are three sequences for the species in BC, and they're all mine, and they're all within 10 kilometers of one another. So it'll be interesting to see if more show up both within BC and also on the West Coast. Next, please. All right, so this is the second one I want to talk about. This is another fungus and fungal parasite kind of situation. This is Russula brevipies, which is usually a white squat mushroom. But this Russula has been parasitized by another fungus called Hypomyces lactifluorum. So the parasite meets the host while still underground as mycelium. And so the mycelium gets infected with the parasite, and when the mushroom pops up, instead of this compact white mushroom, you get this beautiful orange colored, fluted, kind of strange looking species that's known as a lobster mushroom. Um, Lobster mushrooms are very common where I am here in Castlegar, and I wouldn't normally collect one, but this one had been knocked over, so it was already basically detached, and it was just in such good shape, and it was so pretty. So I figured I'll collect it and I'll send it in for sequencing. So when it came back, yes, it had the sequence for the Hypomyces lactifluorum. But it also had the sequence for the host, which was the Russula brevipies. Um, but not only was there Russula brevipies, but it was a particular variety called Acreor. So could I see in the next slide, please? So if you look on the left, that's the hypomyces. You can see it's very widespread. It occurs basically wherever the host fungus is found, which is Russula and a couple of other species. But if you look on the right here, the host species of Russula brevipies variety Acreor is much more spotty. The distribution is sort of down the west coast of the US and the eastern area of the US. And then there's one little dot up in BC. So this is the first time that this particular Russula brevipies, which is a totally common species, but this variety, which is less common, has ever been sequenced in BC. So interesting combination. When I first made the observation, I made it for only the Hypomyces lactifluorum. And the person who ID'd it, who happened to be Kem, who is here and we'll be talking later, he sent me a note and said, "Hey, you got two observations back for this same specimen that you sent in. Can you duplicate your observation?" So on iNaturalist, there's a way to make a duplication of the observation, gives you all the same photos, same map, same date, but you can then call it something else. So I made a duplicate and I called it Russula brevipies via Acreor. So now we have both of these linked observations of the host as well as the parasite. And that was all I wanted to say about them.
Beauty. Um,
Hey everyone, my name is Marshall. I live just outside of North Vancouver here, and yeah, I have a couple interesting finds to talk about. The first one is this Leucoagaricus here, and this one is a BC first. It's never been found in BC before. It's found in southern Vancouver Island by Ronan, and I just chose this one because it has some really nice pictures, and it's a pretty novel find here. Um, so yeah, that's what I have to say about that one. And then here's another BC novelty found by John, and this is Cortinarius angelicianus, and it is called angelicianus because it's originally found near Los Angeles, which is pretty interesting, quite far south. And yeah, I just chose this one as well because it has some more nice photos and it's pretty unique as far as Cortinarius go. Or I guess not unique, but just a bit notable because it has that really cool sort of scurfy cap. Um, and yeah, just a nice find.
sweet. This one's you, Kim.
Okay, yeah, I brought this one along because I want you to appreciate what a pain in the neck this project is for people who write print books on mushrooms. Uh, this particular species, if you look at it, you've all seen it. It's Zoramphalina campanella, and it grows on logs in great clusters. It has decurrent gills. It doesn't have a bitter taste, and We've been calling that for years, but those of us who are sort of on the inside, we knew that there were cryptic species in this, that it was really a species complex or a species group. And that was fine. So we still called it Xeropolina campanella. But about 10 years ago, somebody started to study these things, and as they studied them, they gave descriptions and names to some of the members of this cryptic group. And one of the names they gave it for one of them was Xeropolina enigmatica. Now, we have sequenced this thing 7 times in BC, and guess what? They're all Xeropolina enigmatica. In fact, the only Xeropolina campanella in the strict sense does occur in BC, in on the west coast at least, but as far as we can tell, it only occurs on the tops of mountains and in high latitudes. So basically, anytime you see this in our area, and I live in southern Vancouver Island, anytime you see this, it's very likely Xeropolina enigmatica. So the question is, what do we call it? Do we keep calling it a group, or should we call it what all the sequences tell us it is? Next slide. Next person.
I think next person is you, Cam. You want me to talk about? You're muted again.
Did you want to talk about the BC firsts?
Sure, I can. Yeah. So some of you might have noticed maybe some of your observations, or if you've browsed the results at all, that there's a project that I've made called MycoMap BC First. A bit of an awkward name, actually, but whatever. And basically what we're using this project for is the first record of any mushroom in British Columbia. So that could be described species that match a name or a type of something that we've never had on. This is on iNaturalist, first record on iNaturalist in British Columbia. It could be a temp coded species. So Luke gave a great description of kind of what the temp codes are earlier. I put this X, this is like drama, check, X, but the last one is just saying like, if it's the first sequence specimen, like, we might have a first sequenced, very common Lactarius, but we have hundreds of records of it otherwise. Those ones aren't going in the project right now just to avoid overflowing it, because there's going to be so many things that are the first sequence specimen, simply because we don't have very many sequence specimens on INAT from BC. But yeah, right now there's 62 observations in it as of yesterday. I think it's probably the same now. And as I said before, there's about 210 things that have been processed. So it's a really high percentage. Of course, because we haven't done a lot of sequencing, most things that match a temp code end up going in this project, because it's the first temp coded species. It's the first time we've had that temp coded species recorded in BC. Someone might have collected it before, even had it on INAT, but without sequencing, most people aren't identifying things to a temp code, if that makes sense. Usually we apply a temp code name only after sequencing, although not all the time. But yeah, so I think we'll continue to see lots and lots of mushrooms and collections come back and be first for the province, which is really cool. Like what Kem was saying, there's some things like maybe if we collect 50 more of these things we are calling Xeromphalina campanella, we'll have an even better sample size, and we'll have a better idea of if we really should just be calling them all enigmatica, because that's what they are. So yeah, you can check this out. And then the other thing I wanted to mention is I made a third project, because why not, called MycoMap BC New Temp Codes. So Luke was talking earlier about those 2 inosebes from the Okanagan that didn't match anything well enough to be assigned an existing name or an existing temp code. So they, triggered the generation of new temp codes. So those things are all being thrown into a project as well. Cam, would you like to speak a little bit to this?
Yeah, I thought maybe before we go any further, I'd just like to raise the question of why we're doing this, to sort of get the big picture. And I think you can kind of see why we're doing it when you look at some of the results that have come in so far. We have 13,000 vouchers that are being sent in. Of these, 400 are back, and And as we mentioned, over 200 now are validated. We know what they are. And as Alora mentioned, in the BC First project, there are 60. Now, that's 60 out of, say, 220. So what that means is that 1/4 of all the mushrooms that we are looking at in this project so far, 1/4 are totally new to BC. It's not just that they haven't been sequenced here before, but they are totally new. Now, in BC, we have about 4,000, 3,500, 4,000 known species of mushrooms. And so if you take this out to its furthest extent, and if this project goes on for some time, eventually then we would have in BC an extra 1,000 species that we didn't have before. And some of these will be temp coded, of course, but eventually we hope that even the ones that are temp coded will someday be studied and we'll give names to them. And so really, this project is going to change the face of fungal studies in BC. And I hope this perhaps gives you some enthusiasm for what we're doing and would like to be part of it. When we get to the end, we're going to issue an altar call and see who wants to join. But from here on out, we're down the rabbit hole. So bear with us, where we want to give you an idea of the actual underbelly of what we're doing. But I hope you keep the big picture in mind. Next slide.
Awesome. Okay, so yeah, like Kem was mentioning, we're gonna jump a little bit into the back end now. So this is my silly little timeline that I made. And we've talked a lot, I think, if you've been at the previous meetings about these kind of first three things. So you guys know that people go and collect and dry fungi. They get sent to UVic for quality control. We're doing tissue sampling now. But I want to talk about these sort of next three steps when they get sent to Mycota. And I think if I click, I'm gonna, oh yeah, nice. Get sent to Mycota, the sequence validation, which we've been referencing, and then how they end up on INAT. So to jump into that, it's really awesome we have Alex here because they worked at Mycota Labs and know more about the sequencing process using nanopore and specifically for MycoMap than anyone else here. So I will pass it off to them and they're going to tell us a bit about what this actual sequencing looks like.
Cool. Thank you. Um, I'm gonna try to not be too bogged down in details, but y'all can leave any questions or anything in the chat, and I can get back to you after this. After I'm done yapping, rather. But yeah, as you can see in the slide that's up now, for a while, Mycota Labs was operating basically out of that guy on screen. That's Steven, the founder of Mycota Labs, was operating out of his basement. Not by the time I got there to work there, but by that time we got an actual lab. But for a while, the beginning of the sequencing just was born in his basement as a pet project. And I'm just going to talk a little bit about what the sequencing process is, what it looks like right now, and just how we get all the data that you will find coming up on your iNaturalist observations. So next slide, please. So first step is to get the DNA out of the mushrooms, for the most part. Once you mail over our samples, then they'll get, or once you mail over your samples, rather, they will get recorded into our database. That first picture there is myself and my coworker. We are scanning barcodes at San Francisco State University last summer, recording the accession numbers, just recording the database numbers of a ton of mushroom samples from their herbarium. Now that the MycoMap BC project is using the little voucher slips with QR codes and stuff, the process is going to be pretty much the same. Just scan it all into a spreadsheet. But before they started switching to QR codes at Mycota, then we did a lot more of just looking at the slip of paper and typing things in by hand, which was way slower and way more prone to error. So now we've decided just like barcodes will make this so much easier. And then from your sample bag that has your dried mushroom in it, we would take a little piece off of it. Previously, we would do it with tweezers, just taking like a crumb. And something that I didn't get a good picture of it, but I guess you can maybe get an idea with the toothpick thing here, is something that I think is understated about the sampling process and the sequencing process is like we don't need a lot of tissue. That's why we say we only need a little piece. But when we're pulling things into our little tiny test tubes to do the actual science on them, we only need like a grain of rice, half that, even less. What like the smallest amount we could possibly get is what goes into our test tubes, and the rest of your sample will get stored in just like a big Rubbermaid bin, like a big clear plastic bin, in order and all accessible so that we can go back if we need to. But we only use a teeny tiny bit. And since we only need such a tiny amount for the sequencing, they've been working on developing the toothpick method, which is what those middle two pictures are, of just using a toothpick and kind of jabbing it into the mushroom or on the surface of the mushroom and rubbing it around, getting a tiny bit of dust or tissue or something on it, and then dropping it into a test tube. And that will give us enough tissue to get DNA off of it, which is just mind-blowing to me. That's exciting. So maybe someday in the future you guys will be sending in samples of mushrooms by poking them with a toothpick and putting the toothpick in a Ziploc bag.
Literally, maybe by this summer, actually.
Yeah.
It's coming up really quick. If you
Yeah.
like small things, you might be able to help us trial this by getting those toothpicks in the tubes and for really teeny things. Could be very soon. Sorry to interrupt.
Definitely. I've been talking to Hart Singer, who does a lot of the validation protocols and stuff. Everybody probably knows Hart. If you're in here, you probably are familiar with Hart. But he does a lot of the validation of sequences and a lot of the sequencing himself. And I was talking to him about the toothpick method at a mushroom event that I met him at in California. And he just demoed it for me of how easy it is to just take a mushroom, poke it, and put it in a tube, and then get the results later. And it's crazy. So, anyways, look out for that coming up, I guess. And then we do the extraction part, which is how we get the DNA out of the tissue sample. This is the most sciencey part, as far as I'm concerned, because it's a lot of putting chemicals in test tubes and moving them around and doing stuff to them until you end up with a test tube of clear liquid with your DNA in it. And this process involves a lot of different sub-steps and chemicals that you don't need to know the details of, I guess. If you do want to know them, all of the process is open source. Steven publishes papers about it on whatever that website is called that he uses. I could link it later if anyone cares. But the protocol itself is just basically beat up the tissue so that it breaks into a little dust, wash it with a bunch of chemicals, and then what's left is the DNA that we want. And then we end up with, as the last picture here shows, a bunch of tubes with DNA in them. This is one run of community samples. That's how many's in a run? 960? Just under 1,000 samples per run, which I'll talk about later. But yeah, that's the first chunk. So next slide, please. Yeah, this is the second more intensive sciencey part. So the first picture there on the top left is the primer plates, which is basically just a preset list of DNA tags, like name tags. Each tube of like sample DNA will get its own specific name tag, which is in one little divot in the plate. And then the name tags will let us know that like what the DNA came from, what mushroom it came from, and what iNaturalist record it came from, or rather, it is associated with later. So each sample gets its own little name tag. And then the top right picture is the thermocyclers, which is the main reason why we can sequence anything at all. It's how you get from one toothpick scraping's worth of DNA to something that a computer can pick up and read and get hundreds of reads off of, which I will talk about more shortly. But basically, the thermocyclers are part of the PCR process. I probably should have looked up what PCR stands for because I always forget. It's like polycycle reactions or something. Polymerase chain reaction. There we go. Yeah. I'm such a fake scientist. Anyways, but yeah, polymerase chain reaction, the details is it's polymerase because that's an enzyme that it uses to make lots of DNA. It makes the DNA do different stuff. Chain reaction is that bottom right picture, which shows one chunk of DNA exponentially expanding into tons of DNA. You probably can't read the text on there, but it says after 40 cycles, there will be 2 to the 40th power of DNA, which is about 1,000 billion copies, which is a lot, but that's what we want. Because a computer can't detect one tiny strand of DNA, or if it can, it'll take a while to fish it out of the test tube of goo. So we need to get as much DNA out of each sample as we can. The PCR reactions trim everything that isn't the barcode. So when we're sequencing, we're not getting the entire genome. I mean, we could be if we wanted, but that's a different process. The sequencing that Mycota does and what MycoMap uses is just barcode sequencing, which is just taking one identifiable little snippet out of the middle of the genes. And generally, that snippet is enough to get a pretty good idea of what we're looking at. So the PCR reactions in the thermocycler takes out the little barcode that we want, sticks the primer parts onto it, gives it its name tag, and then amplifies that times 100 billion or whatever. And that's how we get lots and lots of DNA that will actually have a good chance of giving us good data. So next slide, please. There's a lot of extra steps to the cycling process. Again, it's all documented somewhere if you're interested in the nitty-gritty sciencey bits of it. This is the actual sequencing part. So all of that was just preparing the sample for the most part, like getting the DNA into a readable format. That's the top left picture is the libraries. Those are test tubes full of green science goo, which is to say that is all of the DNA bits from that whole, you remember those plates of 960 samples all lined up on the table? We took a tiny bit of each of those and piled them all up into one tube that is green because of the other chemicals in it. So that's green. So that's our library, which means it has a little bit of everything from the entire run. And just pile it into one tube altogether so that we can load it into the sequencer, which is the second top picture there, which I guess there's not really a scale there, but for scale, this thing's like maybe the length of your phone. This sequencer apparatus is like the length of my cell phone maximum, probably less. It's like the size of a vape. I don't know. I don't have a good example, but it's really small. And you put a few drops of that green goo library master mix into the sequencer, and then science happens to it. It sequences it, which is that bottom picture, which is, I don't know how well you can see it, and it's kind of complicated. But the point is that it pulls, that sequencer has a panel with a bunch of little holes in it, and when the sequencer is turned on and running, it pulls DNA through the holes, and as it goes through the hole, it kind of like tick, 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 ticks over the letters, but like with electricity and like chemistry. So it like tick, 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 ticks over each letter, and each letter in the DNA has a different electrical signal so that it looks different, so that the machine knows that there are different letters in there. And that's how it reads it. It's kind of like it's using Braille to like kind of feel the sequence along the DNA, and then it stores that data for later. And that's called nanopore sequencing because it uses the nano-sized teeny tiny pores. And that's what lets Mycota Lab be able to put through thousands of samples at once. Like it runs through 1,000 samples at once. It can do usually about 1,000 a day or more if we hook up multiple of the cells to run. And it's just a little thingy that you plug into your computer, like a USB drive, basically, and it uses the computer's software to process everything. So that's how Mycota can manage to handle that much data and that quickly, is because it's really this cell that is the most like recent advanced technology for handling barcoding data like this. This little cell is what makes it all possible. I just think it's really neat. It's just such a powerful tool to be able to use, and the fact that we have access to the lab, like we as in MycoMap BC participants and mushroom collectors and everything, we have access to like to be able to use this little thingy to get so much genetic data and apply it directly to our real life. I just think that's very neat. So next page, I suppose. I'm going really fast through all of this, sorry. But I don't want to take up too much time. You can tell by meme that says no fear, bioinformatics and data science, one fear, that I'm intimidated by the bioinformatics part, which is basically seeing what the electrical data is that the nanopore cell read and translating that into the genetic sequences that people will post on my iNaturalist observations. There's the computery part of, well, I guess most of the processing, this part is computery. We'll talk more about the validation bit after. But for the purposes of what's done at the lab, there's the base calling and demultiplexing. Base calling is just like matching up the electrical signal stuff with the letters so that it becomes the letters that you know from genetics, GCATs. And demultiplexing, which is untangling the spaghetti of the genetics. Because when we made that green goo tube, that green library, and stuck it into the nanopore machine, it was just all the DNA from the whole thing all mixed together into a big spaghetti. And then the demultiplexing is using the computer program to pull out each strand of the spaghetti and read the label that's on it, that primer that we had put on it before. And then once we have the names of it, or rather, once we have them untangled, then we look at their name tags, we sort them all into piles, all the DNA fragments, sort them into piles, look at how high quality the sequences are that came out. Just like get rid of like broken half-formed sequences that aren't going to tell us anything, things that are obviously the wrong thing that got amplified or sequenced, that kind of stuff. The broad strokes of cleaning up all the data that came out of the machine and figuring and like starting to get names for what we're actually looking at. What's that picture there? That's my the picture on the left is just an example of how the sorting name tags works. So it's like all of these different color pairs would be mixed together in one tube, and then when they're demultiplexed, then they are just put into their piles of like, oh, this color combination means it's sample 1 or whatever, that kind of thing. It's just looking at the sequences and being able to put them, like match them up again with the names of the observations, for example. And then there's my name, and then the bottom right corner. It might be hard to see, but it's not that important anyway. It's just a screenshot from one of the uh, base calling demultiplexing programs, and it says uh, processing sequences match rate 60.4%, and then it's at like what's that 938,000 out of 1,100,000 sequences. So whatever this processing run was, it was pulling out over a million. It was like untangling and sorting over a million sequences at once, and it was about 60.4% accurate in sorting things into the right piles and having usable data, which is really good. Previously, we had gotten somewhere around like 40-50% on some of our best runs, but this was getting about 60.4% matches, which is excellent. So, and then after all of that is done at Mycota Lab, then that data is packaged up and pushed out to the MycoMap servers and out to the databases, wherever they may be. I think they get pushed to MycoMap first and then only uploaded to other databases on request. Like if you are a science person and you use like NCBI databases, GenBank databases, that kind of thing, that's that those only get pushed there on request. Otherwise, Stephen basically holds onto the sequences unless someone else needs them. Holds onto, quote unquote. They're all publicly available on the website. They just aren't pushed to other databases all at once, because that's like millions of sequences at once. But yeah, I don't know what else I might have to say about that. I just talked for a long time about sciencey things. But yeah, if there's any questions, I can take them now or later. Otherwise, I'll happily talk about this at any other time. But
No,
we
that
can move
was
on to
great.
the
You
we can
had
move
some.
on to the next part.
Sure, yeah, that was great, Alex. Thank you so much. We had some questions in the chat, but I think we've kind of Anne and I have been answering them.
Oh, good. Thank you.
Yeah, I love your meme. Bioinformatics are also my one of my several fears now. I've been in what I call bioinformatic boot camp for the last
Yep.
like 6 weeks trying to understand how all this data is processed and sorted. So yeah, I like laughed right away when I saw this in the slides.
It's very intimidating, but you can figure it out. It takes a beefy computer, but it can get done. The easier part for you to figure it out is the verification, the validation.
Yes,
So
which
I'll hand
means
this back off to everyone else.
yeah, sweet. So, we will shift gears here now and talk about what happens after all that. So, our green goo goes into machine, machine produces sequences, sequences go to database, and then they have to get pushed to iNaturalist and interpreted by a human right now. So, I'm going to pass it to Marshall, and Marshall's going to do just like a 5 minute demo of what this validation looks like.
One thing, Laura, it says screen sharing is disabled.
Oh, request. Can you request to do it? It should let you.
It just no, it just gives me OK as a button. I can try
Okay.
to talk through some of it, but a visual aid might be nice.
A visual aid might be nice.
Yeah.
Can you try again? Hold on, I'll try to fix it. Sorry. Share screen. Share button. Sorry, I really didn't think about this. I normally do things all the time in this account, and it always lets other people do it.
If you want to pull up the spreadsheet on your end too,
Yeah, just give me 1 second because I feel like I could fix this, but and then otherwise I'll just do it and you can talk. Yeah.
okay, yeah.
Do you want to try one more time? It wasn't because I was sharing.
Yeah, it's still saying it just gives me like an OK button.
What? Ugh, advanced. What the heck? That's so odd. Okay, I will share and you can talk. Does that work?
Yeah, for sure.
Okay. So I'm gonna get out of the slides, and lucky I have this open, but I don't have everything open, so I better start loading some things. Okay, go for it, Marshall, and just tell me what to do.
Okay, yeah. All right. So hi everyone. Hi again. So I'm going to give you a quick walkthrough of sequence validation. So basically what we do once we get the sequence and how we really use that sequence to eventually lead to hopefully an identification. So the first step is going to be actually finding a specimen that's being sequenced and trying to interpret that. And we do that here at MycoMap BC through this spreadsheet. So this has most of the sequences that have come back to us, and we're all working together. You can see the working field there, and we're all putting our names down when we're going to work on a specimen. So Alora, if you just want to Control F for photoluminescence.
Okay.
Yeah, so I've just selected this one for us to look at because it's a pretty simple one. I'm not going to go into too many details. But could you open the INAT super quick of that observation? Yeah, so basically when I'm going through the validation process, I like to open the observation just to get a look at what I'm actually validating. And then from this page, it's good to grab the INAT observation number as well, even though that can be done from the spreadsheet. And then we're going to take this and put it into the MycoMap BC website. So this web page basically has the same database as the spreadsheet we were showing you in the beginning, except it has the sequences attached as well. So you can see Laura just put in the iNaturalist number of that observation, and then this web page is going to look through the data and it's going to find that and it's going to pull that right at the bottom there. And now you can see that little S button, and that is to look at the sequences. So yeah, if you want to open that, and you can see with this example, you can see sequences there, and then there's sort of like one link. But the interesting thing is sometimes when we get the sequence back, there's like 2 or 3 or 4 different sequences. And this is because sometimes when you send a specimen in, there might be spores of another fungus that you've sent in accidentally. There might be some dirt that's snuck in there or something like that. And it's really easy to get actually DNA of a few mushrooms at once. But like I think was mentioned earlier, the nanopore is pretty good at picking up different sequences of different fungi sort of all at once. So usually, and not always, but usually your sequence will be there as well as oftentimes some non-target sequences. But yeah, if you just want to open that link now, this is going to take us to another page where we can actually see our sequence. So that right there is the sequence that comes out at the end of everything. Now, there's a couple things to mention. The first thing I'll mention is that as you can see right above all of these colorful letters, there's a thing that says reads in consensus 100. And this is because what we're looking at here is actually a consensus sequence, which is a combination of 100 sequences that have all sort of been averaged out to give us this final sequence. There's a lot of variation even within the same specimen of a fungus as to like different ITS DNA codes. ITS is the gene region we're looking at. So different DNA codes from this region that can come out of a specimen. And what we're sort of looking for is a sequence that we can compare to a bunch of other things that aren't so similar. And because of that, we don't want to see the gaps and differences in all of the individual sequences, but a consensus sequence like this. Would you be able to open Eugene?
Yeah, it's gonna take a second. I don't have it pre-opened, but
Okay, if it's a
it's gonna
hassle, no worries.
no, it'll go. It's just slower when you're on Zoom.
Yeah. So what I was just mentioning about the consensus sequence and how we have this one final sequence and then sort of 100 sequences that go into that, there's a software we use called Eugene, which actually lets us look at the 100 different sequences because sometimes it's really relevant to see the individual sequences. You won't always get a consensus sequence with a number like 100. Sometimes it'll be a number like 6. So the individual sequences that are contributing to that 6 reads in consensus sequence are a lot more significant. And then in this software, we're also looking for things like how similar are these 100 sequences to each other, or how different they are from each other. This is called polymorphism. So like if there's a lot of variation, then maybe you can't trust your sequence as much. But if all of the 100 sequences are very similar to each other, then they're probably pretty reliable. You can see it here. These are vertically a bunch of different sequences. And then as you read to the right, you're looking at one individual sequence. And one sequence on top of another is, you can see these are all pretty similar to each other. There's a lot of complicated things we can do in here, and I won't get into the weeds of it because it's very easy to, of how to align everything. But yeah, everything looks pretty similar there. It looks pretty good. So I just wanted to show you this as well. And thanks for opening that, Laura. I think we're good to go back to the MycoMap page, though.
Yeah, this kind of just shows you can see there's 100 rows for all the 100 sequences that have been averaged. Sorry, it took me a while to get this pulled up. And these are all just like little flaws and nanopore errors that once we look at the average, we realize they're not important. But did you want to go back to this one or this one?
That one, yeah, that's good. Yeah, so okay, so now we have our sequence and we've made a consensus out of the 100 original sequences. We're pretty happy with this sequence. What do we do now? How do we actually use this sequence to identify? There's a couple ways to do that from this page, but Laura, do you want to just go to the related blast searches? And yeah, so basically we're going to run this sequence we have against a database of probably millions or at least hundreds of thousands of other sequences and see, especially what we're interested in is the most similar sequences. Because generally things that have very similar sequences to our sequence are going to be closely related, and things that have very different sequences are going to be distantly related. So we pull up this page here, and you can see a bunch of different values, which might be confusing, but the main one we're going to be looking at here is the identity value, and it's giving us a number, and this is a percentage out of 100. So basically, it's taking our sequence and it's comparing it to, this is an entry of another sequence. And basically what it's telling us is our sequence is 99.22% similar to 99.22% match to this other sequence. And then the number below it, 100, is just an adjusted match is what we call it. So sometimes we'll have spaces in our sequence that are ambiguous, which basically means we can't tell exactly what they are. And if they're a potential match to the thing we're comparing it against, then we can say, okay, well, if we adjusted all of the ambiguous characters in our sequence to be matches, how good would the match be? Yeah, so that's adjusted matches. If you want to scroll down to the mycoblast, then yeah, so this is the above was like a GenBank search, basically. A GenBank is just another database. And then here is like a mycoblast search. And this is going to give a different database. It's supposed to be, I guess this one's supposed to incorporate the GenBank search as well, I think, for type specimens, but it's good to look at both. And yeah, so the top entry here is actually our sequence. You can see that our sequence is a 100% match to itself, which is good, because if it wasn't, something would be wrong. And then if we move down the list, we can see that there's lots of close matches and that they all have the name Pholeotobernescens. And if we look at this list, we can be pretty sure immediately that, okay, if we compare our sequence to the most similar sequences and they all share the name Pholeotobernescens, even if we didn't already know the name of what we're searching with, we can be pretty sure that it's the same thing as all of the things it's most similar to. In this case, it would be Pholeotobernescens. There's one more wrinkle in the process, which is just because all these things are called Pholeotobernescens doesn't necessarily mean they are that. I mean, in this case, we have like 20 or 30 things called that, so we can be pretty sure. But I'm just mentioning this because it's good to look for something that's backed up by a bit of evidence as well. So something like a type specimen is really good if you have a sequence of a type specimen, which is a specimen that's directly like linked to the uh, to the name itself. Um, and in this case, we do. So you can see the third closest match here is a type specimen of Foliota. And if that's closer than any other match of any other species, then that's pretty much all we need right there to say, okay, this is Foliota. So I'll usually look around this page and find some links and then go back to the INAT. And then now that we're pretty sure this is Foliota, what I would normally do is leave a comment saying Foliota 100% adjusted match to the type in Oregon. And as you can see, Alora's already done that because she was the one that actually originally validated this sequence. And as you can see, it's the first record from BC. So that is more or less the sequence validation process. If you have any questions or if I missed any, feel free to ask.
Sweet. Thanks, Marshall. Sorry the screen share didn't work, but that actually worked pretty well.
Great, yeah.
Yeah, so that was like a very brief overview of kind of what we're doing behind the scenes so that you end up seeing like this name on your observation. There's this human element of somebody analyzing these results, analyzing the reads we saw in Eugene, and just sort of basically making sense of all of this and applying the best possible name. Stephen of Mycota Labs calls this a preliminary triage, i.e., some of these names in the long term might not hold up. Other types might get sequenced, things might get merged together, they might get split further. But basically, what we're doing, like, this is a bit repetitive of what Marshall was saying, but yeah, just trying to find the best possible name we have right now, and try to do a little investigation into that. And we're going to be talking about that later because it's something that we need more help doing. Kem mentioned we have 400 sequences back. We've been working away and we've gotten through, 220, 230 in the last month-ish. We're being very thorough, to be honest. I think we're doing a great job, and that's why we're kind of like a bit slow at it. But the more people we have, the more we can get through. And keeping in mind that that's 400 and we have like, another 12,000 something coming down the pipe, like it's going to be nuts. So I'll leave that there for now and go back into our slides because we have one last thing to show tonight. We kind of did this. It's just like a little analysis of understanding identifications, comments, what do they mean? So a little bit about like the terminology you might see. You collect a mushroom, it gets validated, someone comes and leaves a comment. How do you interpret that? So, I don't remember exactly who was going to speak to each one here, because I can't see my notes. Leanne, this is
That's
you.
me.
Perfect.
Yep. All right. So I was going to talk about the comments on this observation here. This is a collection by Ronan, and it was identified as Lactarius lucentus, variety lucentus, luculentus. Okay.
It's a mouthful.
I always tell my students that it doesn't matter how you say the names as long as you pronounce all the letters. Lactarius luculentus, variety luculentus. All right. So the variety means that it is a subspecies. So varieties within a species are slightly different from each other genetically and will have a slightly different but consistently different appearance or qualities that will be able to differentiate them. There are two varieties of Lactarius luculentus. This one is the less common variety. It can be differentiated in the field, but it has a duller cap color and a slightly more acrid taste than the more common variety, which is variety latus. There's a little star there beside the word type specimen. Marshall talked a little bit about a type specimen. It's the mushroom collection that the type was originally described from. So that's type specimen. It says here it's the second sequence from BC, so this sequence has been found before, and it gives examples of two other observations here. The other two observations in BC was 2003 on Salt Spring Island by Vail, and then this one here, which Ronan found near Souk. DNA barcoded observations that are linked here, those two iNaturalist links that you see there, were both from 2025 in Washington State on the Olympic Peninsula. So there's another one in BC, but these two linked observations were more recent. They're a couple more years more recent, and they're also quite close to us, or quite close to Souk and Salt Spring Island being on the Olympic Peninsula. All right, so the next one, please. All right, so we looked at this one already. Luke talked about this collection by Mitch of Folioto Kodakiensis, collected by Mitch Milgram, 100% adjusted match. Which means that the barcode that we came up with from the specimen here matched absolutely perfectly the observations that are linked there from Alaska and Alberta. Once what's called ambiguous nucleotides have been accounted for. So sometimes within the DNA of a particular species, there are certain standard variations. So there'll be one sequence that's almost exactly like another sequence, but they both occur within this same named species. So that's what's called the 100% adjusted match. It 100% matches one of those common variants. So the species that were chosen there from Alaska and Alberta were chosen because they bracket us geographically, right? We have one to the north of us in Alaska and one to the east of us over in Alberta. And then it says that it's a 99.86 adjusted match to type match. So just from my experience of knowing how many letters are usually in one of these ITS sequence, 99.86 adjusted match means that there's probably just one letter that is different between the example that we have here collected by Mitch and the type species that was collected in Alaska. So probably one of those is one of the variants and one of those is the other variant. So we have them, one of them represented here and the other one was represented in the type collection. So that's what the 99.86 adjusted to type means. So generally any match over 99% should be considered a potential good ID. Any match below 99% is most likely, most times going to be a different species. So you're looking for something over 99%. So I didn't look at this barcode myself for Foliota kodakiensis. But it's so slightly different from the type species. Kodakiensis, the name was given because this was found in, the type species was found in Kodiak, Alaska. So that's kind of nice that the Latin names tell us something about the species itself. So down below here, it says, below the type match, it says that there's a number, an alphanumeric MZ05, etc. Then there's the name of the species, where it's from, the fact that it's a type, and another set of numbers. So that first set of numbers is the NCBI accession number. And an accession number is a reference number that's given to any specimen when it's accepted into a herbarium or mycarium. NCBI is the National Center for Biotechnology Information, and it hosts a database of publicly accessible genetic sequences that's known as GeneBank. And Marshall mentioned GeneBank earlier. So if I wanted to find out more about this excuse me, the type specimen that's mentioned in this comment, then I could go to the NCBI webpage and enter that MZ05 number into their search bar, and it will bring up the record for that type specimen. It'll tell us things like where and when the type was collected, the collector's name, which, could be any of us if we're collecting something new, and stuff like that. So that's where that NCBI accession number can come in handy when you just want to find more about the cool mushroom that you found. The number following the type info is the isolate number, and I'm not sure what you can do that with that. It refers to the identity of the pure tissue culture of this Pholiota kodakiensis, and I don't think we really use that in the validation, but there's that number as well. What else? Okay, and then the novelty in the region. So this is considered to be a first for BC, although we do have the Alaska and Alberta collections. So when we talk about regional novelty, it's based on DNA. So a lot of people may have said, yeah, I found Pholiota kodakiensis in BC, but unless it's been DNA sequenced, it's kind of just a rumor. You may have found something that looked like Pholiota kodakiensis, but to definitively be able to identify it as, yes, it's here in BC, you need the DNA. iNaturalist has something that they call a research grade observation, which is when more than one user agrees, yes, this looks like this species. And it could be correct in their agreement, or they could be completely incorrect. So until that DNA is actually found within the province for the first time, it's not confirmed as being here. So now that Mitch made this collection and it's been sequenced and it's been found within BC, then it's definitively, it exists here.
Sweet.
And we already talked about the MycoMap BC First project, but this would be one of the mushrooms that is included in that project. All right, that's all I wanted to say. Thanks.
Nice, thanks, Leanne. So yeah, Leanne mentioned type sequence. So essentially the sequence derived from the physical mushroom that was originally used to describe that species, which is ideal scenario. But unfortunately, a type sequence isn't always available. We've been describing mushrooms for a couple hundred years now, but sequencing them for far less. So the described mushrooms outweigh the number of sequences that are available. But when we're looking for evidence to support through our validation process, you're kind of looking at as many different places as possible to kind of gather and build a case for a name you're about to assign for it. So in this case, there was no type specimen available, no type sequence available for this Leucopholiota lignicola. But while I was looking through all the connected pieces of information around this mushroom, I did locate a paper from 2022 that sequenced collections of Leucopholiota lignicola from mushrooms picked in the same area of Finland of where this mushroom was described from in 1879. So you can see here we have a comment that says 99% match to type locality sequences, and then there's a link to those GenBank sequences as well. So this can be referred to as a type locality sequence, and maybe doesn't carry necessarily as much weight as a type sequence, but it is just one more thing to kind of justify the name you're putting on it.
Sweet. I think I'm just blasting through the rest of it, or
you
with this one?
I think, yeah. So basically this one is a good example of how there's not always a definite yes or no answer as to whether a temp code should be used. You can see in this case we have Lyophilum decastes IN01. It's 100% match to validated sequences from North America. But what's interesting, if you want to go to the next slide, is that there's a very similar mushroom, the true Liophyllum decastes, in the EU, which actually varies within itself by about 1% in ITS. And when we look at our North American sequences, we see that they're all usually about 1% different. So it's sort of just on the far end of this variance that we see within the true Liophyllum decastes. So it becomes a question as to whether we should call our mushroom Liophyllum decastes or whether we should give it the temp code. Um, I guess in my opinion, I would lean towards giving it the temp code because we don't see all of the 1% variance in North America. We see them all around an entire 1% different. But there isn't really a definite answer, so it's just an interesting thing, and this happens pretty often with lots of different mushrooms.
I'll unmute myself. Would be a good idea. Um, Yeah, so I'm just going to go through the last couple ones here. These are just more examples of how to interpret the comments you might get on iNaturalist. Here's an example of something that doesn't have a type. There wasn't a type locality to reference. So we're referencing instead to other observations that have been given that name, that are sort of starting to build up like a concept of the species that could change in the future. And then you also might see reference to a database called Unite. So Unite is a European database that stores sequences mostly for fungi, and they're all automatically curated based off like barcode gaps and percentages. And then they have what they call representative and reference specimens for certain species that you might find that your mushroom is a match to. So it's not as good as matching a type specimen like we saw, but it can help point us in the right direction. So if you see Unite, that's just that European fungal database with sequencing info. And then some other things that you might see, we've seen by now, the GenBank or NCBI or accession numbers. You might see references to, if you see, I've been writing this all the time, see Danny's notes, see Danny's notes. Danny is Danny Miller, and his website, this is a link to something on his website here, has a ton of info about mushrooms that have been sequenced in the Pacific Northwest. He's been at it a long time and knows a lot. So often I'll consult his website to see if something may be the best name or what I can learn about it. And then sometimes I'll reference as well Index Fungorum. So Index Fungorum just has this big list and databases of all the names of described species, and it tells you often where they were described. So was it described in Washington? And then that's helpful in sort of trying to discern if we might have that in BC. But sometimes it was described like we saw in Finland, and it will still be a match, but it's just another tool to basically learn more about the mushroom we're looking into and try to discern if that's what our collection is. Okay. I lost my video bar here. I don't know where. I'll bring it back. Oh, did I end the screen share or did I just do something silly? Oh well, I won't see you guys. So we have come to the end of our meeting here, but there are next things for people to be involved in if they wish. So we've decided, and we is like me and pretty much everyone else who spoke tonight, that on February 3rd at 7:30, we will do a deeper dive into the sequence validation process. So what Marshall demonstrated, where we ran the blast search, we looked at the reads in Eugene, and basically this process of discerning what is the best name that we know of to apply to our collection. How do you do that? We need more people that want to do this, that want to dive into the data a little bit. So if that is you, what I will do is I'll just send everybody who was on this meeting an email, and I will send like a spreadsheet or ask you to reply or something if you're interested in learning a bit more. And if you're free on the 3rd, you can join us. It won't be like a presentation. It'll be more like just demonstrations and conversation and stuff like that. And then after that, there'll be some more steps. So Fundus, which is an organization out of California, for the whole US really, has put together some resources related to how to do this. And there is a working group for all of North America that meets pretty regularly that you can join and learn about this stuff. So, anyway, I think the most important thing is if you want to learn how to do this and help us go through BC data, anyone can learn it. You don't need like a biology background. You don't need to have like fantastic mushroom ID skills. There are ways that sort of everybody can gain the skills needed through um, time and practice. And yeah, if you're interested in it, you'll probably be able to learn it relatively quickly. So look out for that email if you would like to join. We would love to have you. And then afterwards, we can hook you up with these other resources mentioned. Um, but yeah, that is kind of our last slide. So I'm going to stop my screen share. And if people do have questions, we can take questions. And otherwise, um, thank you for coming. Hopefully, you learned something about um, this process behind the scenes. And yeah, that's it. So feel free to head out or speak up if you want to ask something.
You guys have been doing so well at keeping up with the questions on the chat.
Yeah, there's been a lot actually.
Fantastic. Mhm.
All right. Well, maybe we're good. Okay, well, thanks everyone.
Thanks, everybody.
Thank you.
Have a good evening. Enjoy your weekends.
Yeah, thanks everyone. Bye.
Bye, everybody.
Thank you. Good night.